So I think this video is probably going to be one of the longest ones I've done so far, and in it I'm going to be talking about some features of prokaryotic cells, and to start we'll look at the glycocalyx. So this is sort of like an outer membrane on some cells. You can either have a slime layer, which is loosely attached to the cell, or you can have a capsule, which is a tight matrix attached to the cell wall. Both of these allow for it to grow on surfaces and offer some protection. Next up we have the flagella, which is a long filamentous appendage which aids in locomotion, which is movement. And wish me luck in saying all these next couple of words. So there's some like different types of organisation that they can have. You've got patricious, which is when they can be all over the cell. Polar, which is when they're at either ends of the cell, so you can have monotricious, which is when you've got one at one end. You can have lopitricious, which is when you've got quite a few at one end. You can have ampitricious when you've got one at each end. And then you've got atricious, which means it doesn't have any. Now it's important to remember we're talking about prokaryotes now, because prokaryotes and eukaryotes use the flagella to move differently. And in your prokaryotes it uses rotation for propulsion. And then if we look at the actual structure of the flagella, you'll see that it's actually made up of flagellin protein, which is in a helix around a hollow core. It has a wide hook and a basal body, which actually anchors it to the membrane. But how it does this varies, whether it's gram-positive or gram-negative bacteria, but I'll talk about that a bit later on. And then we'll go on to some similar things to an actual flagella. So you have an archaea, which is classed as an analogue of the flagella. This basically just means it's similar to it, apart from it's narrower, it lacks a basal body, and it is believed to have evolved from the flagella, which is why it's obviously similar to it. You have your fimbriae, which are filaments again, and they can be at the poles or all over, but they can vary in the number from like 1 to thousands, and they aid it in binding to surfaces and other cells. So like it's when you get biofilm over water or liquids. And then you've got your pillae, which are longer than your fimbriae, but they're a lot less in number, and there are two types. So you have your conjunction or sex pillae, which allow for transfer of plasmids, which are like the little circular DNA, or you have type 4 pillae, which allow for twitching motility along surfaces. So you can imagine it's sort of like hopping along or like squirting along. I don't know what that term is, like shuffling along like a little caterpillar. And then we're on to the cell wall, which I used to love learning about for some weird reason. So not many species, but some species of prokaryotes actually do lack a cell wall, and instead they have a tough outer membrane like the glycocalyx that I mentioned earlier. And for your bacteria, the cell wall's made out of something called peptidoglycan, which is a polysaccharide. So it's made out of monosaccharides, and these monomers difficult to say, so they're abbreviated to NAG and NAM. These have a beta-1,4 linkage between them, and a whole backbone consists of around 10 to 65 sugar layers, which can form cross bridges between them. They can also have something called a tetrapeptide side chains, which link multiple layers together. And then I mentioned earlier about gram-positive and gram-negative, so let me tell you about that. This is because of different structures within the cell wall. So for gram-positive, it has a thick peptidoglycan layer, so it retains something called crystal violet dye that we add onto it. And then when we add a secondary stain on called saffron, because the crystal violet is still present and it's such a dark purple, it masks the red colour of the saffron. So gram-positive bacteria you see is purple if you sort of stain it with crystal violet because of that thick layer. Whereas with your gram-negative, it only has a thin peptidoglycan layer, so when we wash this crystal dye out, it washes away, and the crystal violet stain is taken up and stays visible, so your gram-negative is seen as like a reddy colour. So if you just sort of think about it, you've got your gram-positive basically has 2 layers of peptidoglycan, and your gram-negative has 1, because one's thick, one's thin. So now we've covered one type of prokaryotic cell wall, we'll go on to the other, which is your archaea. These have a lot more of a variety of what the cell wall can be made up of, but it's never made up of your peptidoglycan, that's only your bacteria. So some examples of what it can be made up of are pseudomyorin, which again has some very, very difficult to say monosaccharides, and I'll let you try and pronounce them. And these are connected similarly, but they have a 1,3 beta linkage instead of a 1,4 beta linkage. Your most common type is something called S layers, which are a paracrystalline surface of interlocking proteins and glycoproteins. Now these can be things that make up the cell walls themselves, or they can be cell walls of S layers and something else. And then now we're going to go on to another one of my favourite topics to talk about, which is your cell membrane, or which is also called your plasma membrane or your cytoplasmic membrane. This is a type of phospholipid bilayer, or at least a bilayer made up of phospholipids. This is a type of lipid where it has a hydrophilic head and a hydrophobic tail. Hydrophilic meaning water loving, hydrophobic meaning water hated. It allows the cell contents to be separated from the environment. It allows for molecule passage in and out, and it's also a site of some metabolic processes such as respiration. And there's only a slight difference between prokaryotes in this. So in your bacteria, you have phospholipids with an ester linkage, whereas in your archaea, you have an ether linkage there, just to make it a bit harder, give it very similar names. It's often described as a fluid mosaic because it's fluid, because it can sort of move about, and a mosaic because it's not just made up of these lipids. It can have things like cholesterol, ion channels, carrier proteins, channel proteins, glycoproteins, glycolipids, all sorts. And it's through some of these channels that are in the membrane that movement can occur. So you can have active transport or active movement. So your active transport is when you move molecules against a concentration gradient, which means it requires energy because you're moving it against how it wants to flow. So that uses ATP. And then you've got sort of the reverse, which is your passive transport, which is high to low concentration. This can either be done by simple diffusion, which is when you've got like small molecules which can move directly across the membrane, or facilitated diffusion, which uses channel or carrier proteins. So this is like your ions and stuff. And you've got your specific type of movement, which is a movement of water via osmosis. This is from a high to low water potential by a type of protein in the membrane called aquaporins. You've then got that thing that I like to think of as like jelly, which is your cytoplasm and your cytoplasmic matrix. This is actually 70 to 80% water, which is why it's often seen as like transparent and sort of thick. It has a cytoskeleton which helps it maintain shape, aids DNA replication and division, and until recently it wasn't actually thought to be in prokaryotes. And then in your cytoplasm you've got something called a nucleoid, which is a piece of like long circular DNA that isn't membrane bound, unlike a nucleus in a eukaryote. So because of this, in your prokaryotes, it's not at all associated with histones, but in your archaea, it may be associated with some histone-like proteins. And then you may remember me mentioning these regarding the conjunction pili. There are these things called plasmids, which are bits of extra chromosomal DNA in the cytoplasm. These are around about 5 to 100 genes long, and they can act as a selective advantage because if these have like the gene for antibiotic resistance and they're passed over via these conjunction pili to another bacteria, then that bacteria then has the gene for antibiotic resistance, which can pass on to the next and the next and the next. And an example of when these have been used for good though is in biotech, because we have used them anyway to make insulin. And then we're going on to our little hamburger, aka the ribosome. So in your prokaryotes you have 70S ribosomes, which are used for protein synthesis, and you can have like tens of thousands in the cell. This is really annoying, but it's called a 70S ribosome, but it's actually made up of two subunits of a small 30S subunit and a large 50S subunit. And you now know, hopefully you know that 30 and 50 does not equal 70, but it's just because scientists are mean. So the base of these things called Svedberg units, which is what the S stands for, and that is the rate of sedimentation when they undergo ultra high I can't say this word either. When they're put in a centrifuge at really high speeds. So when you combine two things, the shape changes, and because the shape changes, it means they have a different rate of sedimentation than what a 30 and a 50 added together, like without a shape change, would. So they have a different rate, which is 70S instead. And then we've got some things that vary that by like cells and functions. So they're called inclusions, which act as like a sort of storage inside the cell to stop the effect on osmotic pressure. So you have metachromatic granules, which are sort of inorganic phosphate stores so that you can make things like ATP. You have sulfur granules, you have polysaccharide granules, which act as starch or glycan storage. You have carboxysomes, which contain your ribulose 1,5-biphosphate carboxylase, which acts as a carbon dioxide fixation, which is carbon fixation. You have gas vesicles, which help in positioning in water columns. And then the best ones, in my opinion, the magnetosomes, which help orientate them to the Earth's magnetic field to aid them in finding favourable conditions to survive. And they may also be involved in hydrogen peroxide decomposition. And while we're talking about survival, to finish off, I'll talk about some endospores, which uses another fantastic word and they're formed through endospoilation. This is studied in pathogens such as bacillus, and they can have sort of different morphologies. So you've got terminal endospores, which happen at the end, subterminal, which are a bit before the end, and then central, which appear obviously at the centre. So if you look at how these form and what they do, they occur in some species when conditions become unfavourable. The chromosome duplicates and separates and becomes encapsulated in some things called spore layers. When the conditions become so unfavourable, the vegetative cell dies and the spores sort of released, but it doesn't germinate straight away or live straight away. It can lie dormant for a long, long time. And then when the conditions become favourable again, it can germinate and start its life. At least start its life again, because remember, this isn't the type of reproduction because the number of cells stays the same, so you're not getting two new cells, you've just got one the same, so it is not reproduction. Also, a little tip to remember is what my lecturer called it. It's basically a genome escape pod, so you can sort of like imagine it like it in Megamind, the bacteria just sort of putting its little genome into a little space capsule and shooting it off into space to protect it. I like how my mind went to Megamind and not Superman. Anyway, so that is all I'll cover in this video. It has been quite a longer one, so hopefully I didn't bore anyone or bore myself, because I'll probably be the one rewatching these for revision. But anyway, see ya.